Okay, let's going to keep on going as we move into this next major section in the course. We're going to now be talking about more of the functional side of the nervous system. We have previously covered some of the basic anatomical features, both at the cellular level, the anatomical structure of neurons and the function, of course, of neurons, as well as some of the larger anatomical structures of the central and peripheral nervous system. But now we're going to get into some of the more details about how each of these subsystems, primarily now focusing on the central nervous system, although we're going to involve some sensory components. So we have some aspects of the sensory input to the brain, but we're going to quickly move into what's happening in the brain. And we're going to start with vision. Now, we're going to cover all of the senses at some point over the next few chapters here. But vision is one of those senses that has received by far the most attention when it comes to both psychological research to understand the nature of sensation and perception, but also at the level of neuroscience research to understand how eyes work, but not just eyes, of course, but what's really going on in the brain here. So let me see if I can move ahead there. All right, so we're going to start by talking about the basics of how the sensory systems work. Sorry about the dog barking there. And we have to understand this term called transduction. This is something that actually happens for every single one of our 5 senses. If we just want to use 5, we can debate over how many senses we really have, but transduction is the idea that it's defined as converting energy from one form into another, right? But specifically, sensory transduction involves detecting some form of energy in our environment and then responding to that, but converting it into a different form. In particular, of course, converting it into neural energy. So the receptors for vision are called photoreceptors. And we're going to talk some about those in a minute, but that just means light receptor, right? Photo means light. And so light is a form of electromagnetic energy, but what these do is they respond to light energy and convert it into a neural signal, which is then processed and transmitted to the brain. And so, of course, hearing responds to the pressure waves generated by a sound producing event and transduces that into nerve energy as well. The sense of touch responds to mechanical energy pressure on the skin and transduces that into neural energy also. So in each one of these cases, we're going to talk about the actual point of transduction, but then what happens when that signal is transmitted further into the brain to enable further processing. So for vision, we're going to start with photoreceptors, and then we're going to talk about how the visual pathway takes us from the eye to the brain and what kinds of steps are involved along the way and what we might learn from this. And when it comes to understanding how we generate perception, that's really the goal here. How do we perceive our world? We can approach this from an experimental psychology perspective, but we can also approach it from a neuroscience perspective. How does the brain support our ability to look at an object and recognize it, to look at something and say, that's a cup, that's a table, that's a dog, and so forth. But also we have additional things we do with vision. Vision is one of our key spatial senses. We can perceive depth and where things are and how they move. We also can perceive ourselves visually, not just, of course, by looking at our body, which is potentially obvious, right? Of course, that means when I'm reaching for an object, I can see my hand approaching the object and I can guide my hand into place. And so this idea of using visual feedback and visual guidance of movements is an important thing we want to understand. But also something more subtle is involved with vision, which is when I am walking, even if I don't see the rest of my body, like I'm not looking down at my feet when I'm walking, most likely, right? I'm just looking straight ahead. So I don't really see my body directly, but what I do is I see the visual consequences of my own walking, right? So as you step forward, you start to generate perceptual motion. If you're walking through a stable, stationary room, for example, and you have a bunch of objects that are just sitting in a place, as you start walking past all of them, you experience visual motion as those things either get closer to you as you walk towards them, or maybe they stream off to the side of you as you walk past them, right? This is called optic flow. But the idea is that these visual consequences of our self-motion, optic flow, is also actually useful for us when it comes to controlling how we are walking in the environment. It provides information that we can use to guide our movement. But now let's just start with the basics of detecting light. That's the first step. We need receptors for light. As I already noted, light is a form of energy, electromagnetic radiation. In the entirety of the universe, there's a lot of different wavelengths of electromagnetic radiation that include things like microwaves and gamma waves and infrared light and radio waves and all these sorts of things. But there is a very narrow band of this spectrum of light that ranges from about 400 nanometers to 700 nanometers, give or take a little bit on either side of that. And so that nanometers refers to the actual length of the wave of the light that we're talking about here. That represents the visible spectrum. And this means, of course, that these are the wavelengths of light that the receptors in our eyes can detect. We cannot see gamma rays or microwaves and ultraviolet and things of that nature. Nature has, of course, also evolved many different solutions to light sensing. There are different kinds of eyes. For example, I have the simple version here of just most vertebrates have what are called a chambered eye. Also, some other non-vertebrates have it as well, like squids. This is an example of what's called convergent evolution, that two different unrelated lines of evolution converge to the same solution to a problem. The problem meaning, how do I sense light? The solution is evolve a chambered eye. But the chambered eye in squids does not share a common ancestor with the chambered eye of vertebrates. The definition of a chambered eye is that it contains a light-sensitive layer, the retina, contained within an enclosed chamber, which will, of course, be the interior of your eyeball itself. Different is what we find in many invertebrates, the compound eye. And the compound eye is what you see with insects, a collection of tiny, if you look up closely at it, you'll see that it's actually a collection of a bunch of individual, I call them lenses here on this screen. They're technically called ommatidia, and they're really long cones that project down into a central bulb. And each one of these long cones contains a single light-sensitive neuron. And then they have axons that all collect down at that bulb base at the very bottom of the compound eye. So this is sort of like many different eyes, each one detecting light coming from a very specific direction, and it all gets integrated somehow by the insect nervous system. I threw this picture in here just as a reference point that we want to see. As I said, there's all these other wavelengths of light that we cannot see, but we see that very narrow band of the spectrum from 400 to 700 nanometers. There's a couple of interesting notes here, for example, about how the eyes of some insects, for example, are capable of seeing into the ultraviolet. So that's kind of an interesting thing to make note of here. But for the most part, we're going to focus on human vision here. I already commented about transduction, so perhaps I can also not linger too long over this particular slide, but the idea is that vision is about converting light energy into a nerve impulse. Hearing, I'm calling it mechanical energy here, because in a way, when you have a sound producing event, like if I clap my hands like this, what's happening is I am compressing the air molecules and I'm creating a pressure wave through the air. And of course, that's a mechanical compression of the air molecules. And so that's a form of mechanical energy. We could also call it just a pressure wave. And likewise with touch, when you are touching something, it comes into contact with your skin, and then you have mechanical pressure directly on the skin. And there's also some stretching and pulling of the skin that happens with your sense of touch. Separately, your senses of taste and smell. And we're not going to have a whole lot of detail on those two senses, since those two are the ones that are really not studied very well. Those are called the chemical senses, because the receptors here respond to different kinds of chemical in foods that we may eat or things that we inhale. In fact, one way to think about this is that the responsiveness of these particular receptors, taste buds in the tongue and the olfactory receptors in the nose of the olfactory epithelium, it works a lot like neurotransmitters. So if we remember back when talking about the synapse, I compared a neurotransmitter as a key that opens a lock, and the lock is the receptor on the dendrite that only accepts molecules of a specific shape. And so some receptors will respond to serotonin, but not to dopamine, because those two chemical neurotransmitters have different molecular shapes, and only one of them can unlock the receptor and trigger either an ionotropic or metabotropic response in the postsynaptic neuron, right? Well, the same kind of stuff is happening when it comes to taste and smell, is that the different chemicals, either in food that we're eating or anything that's in the mouth coming into contact with the taste buds, or anything that you have inhaled coming into contact with olfactory receptors, has a specific molecular shape. And some of the receptors will respond to that because that molecule can bind to that receptor and activate it, activating the cell. Or they won't, right? And so in this case, it's a direct neurotransmission, synaptic kind of response, really. All right, so let's get to talking more about vision, as we need to do here. So we're going to talk about first the photoreceptors, right? The light receptors. And we want to ask a few different questions about them and how they work. So things that we're going to think about as we go through here. Receptor density refers to the fact that when we look at the inside of the eye, the retina, which is the lining of neurons on the inside of your eyeball, it is not uniformly dense. There are some receptors that are very closely packed into a certain region of the back of your eye, and that's called the fovea. And in particular, in the center of the fovea is called the macula, or macula lutea. And that's the most dense concentration of these receptors. There are also, we're going to find at least two different kinds of receptors. The simple version is just rods and cones. We might find that there's subtle variations of different kinds of rods and cones, for example, specifically cones. But rods and cones, there's at least two different types there. But we also need to ask these other important, more functional questions. When light activates the rods and the cones, how is information about the light encoded into a signal that goes from eye to brain that provides information about the world, all right? So if you think about the properties of objects that we can see the color of something, right? I can tell if it's red, green, or blue, or yellow, and so on and so forth, right? So the question is, how does the activity of these rods and cones provide information to the brain about that color? I'm also pretty easily able to judge the shape of an object, right? Humans are very adept at distinguishing squares from circles, from triangles, and all other more complex shapes as well, of course. So how is that information encoded into the signal that goes from eye to brain? What kind of, pattern of activity across these receptors can signal to the brain information about the shape of something? And likewise for size and distance. These are more spatial properties of something. How big is it? How far away it is? These both might involve some sort of metric measurement of the space, right? Whether we perceive something in some sort of unit, such as inches or centimeters, which is essentially unlikely, but it might be possible that we perceive the spatial scale of the world in what we call body scaled units. So for example, and when it comes to reaching to grasp something, I'm going to grab a pen real quick here, right? So if you see me on my little screen here, the idea is I might be able to perceive this pen in some sort of a grip scaled, how do I want to say it? Grasping or grip scaled unit, right? And that could be important for providing information about how do I grasp it? If I can perceive this in some sort of hand-sized units, right? I see it in relation to the size of my hand. Maybe that's one way in which we understand the scale of the environment. Direction as well is something to the left or right. How is that coded? And also how, if an object is moving, of course, I can distinguish between something that's moving and not moving, but how do I do that? And this is kind of tricky in a way, because if you remember a minute ago, I was talking about when we are walking around the world, and maybe everything is stable. You're walking through your house and there's a couch and a table and a chair, and everything is just sitting in place. You nevertheless experience visual motion, right? But if you were standing still and all those things started moving, you would also experience visual motion. So how in the world do we manage to discriminate times when we are the thing that's moving and the external world is stationary? And discriminate that from the time when we are stationary and something external to us is moving. And what is most often the case is that lots of things are moving. I might be walking around in the world. Some things are stable, but other things might be moving. So I have visual motion happening in the eye and in the nervous system. Some of that corresponds to self-motion. Some of it corresponds to object motion. How is the brain able to tease apart those two different sources of motion to correctly kind of assign the movement to the appropriate object, right? That's kind of tricky. Not to mention the fact that we can also judge things like the direction of motion, the speed of motion, right? We're pretty good at discriminating velocities, maybe not so good at discriminating accelerations. These are all things we need to want to understand and figure out how the nervous system is capable of extracting that piece of information and encoding it into a nerve signal that is then processed somehow in the brain so that we can consciously perceive that. That's not trivial. These are the major, big, significant problems when it comes to understanding vision. So the simple way of stating the problem is in this next point here, that somehow we experience visual sensations. This is when light activates these photoreceptors on the retina, and those cells are activated in response to some property of the light, such as maybe how intense the light is and what wavelength of the light is. And so we're going to get some differential reaction from those photoreceptors regarding those properties of the light, but then some pattern of activation across these receptors is sent to the brain, and somehow this generates conscious visual perception of the what shape, what size, what color, what distance, what direction, what movement. All of that stuff comes out of there somehow, right? Keep in mind that one of the things that we will need to remember when it comes to how neurons can encode information is that their firing is essentially all or nothing, right? Remember, an action potential, we start at a resting membrane potential of -70 millivolts, and we might get some graded potentials that come in, but if those graded potentials are below the firing threshold, the neuron doesn't do anything, right? But as soon as we cross the neural firing threshold, we end up with an EPSP, and we get an action potential that spikes, right? Now, one thing that we need to realize here is that even though the neuron fires an action potential that looks exactly the same every single time, so there's no such thing as a weak action potential versus a strong action potential, right? There's just, does it fire? Yes or no, right? One thing that can happen is that neurons can either increase or decrease their rate of firing. So as it turns out, neurons have what's called a spontaneous firing rate. And what that means is that because occasionally potassium and sodium might be leaking in and out of the neuron, and we talked a little bit about that in a previous chapter, you get spontaneous action potential sometimes. Sometimes enough sodium or potassium leaks in and out of the cell that the sodium-potassium pump cannot keep up with that, and you're going to get random spontaneous action potentials occurring in neurons. So this is called either the spontaneous neural firing rate or a background or baseline neural firing rate. And what we might find is that if we start receiving some excitatory signals here, EPSPs, we're going to see the neuron not just firing an action potential, but what it really does is it increases its firing rate relative to that baseline, right? As a general rule, most neurons can fire about 1,000 times per second at a maximum of their firing rate. But of course, they can also go anywhere slower than that. If the neuron is receiving an inhibitory potential, that's an IPSP, right? That might actually decrease the neuron's firing rate. So not only does it not fire an action potential when it's inhibited, but it doesn't fire any of those spontaneous background baseline accidental action potentials either, right? And so in that case, that background firing rate, spontaneous firing rate decreases towards 0, right? So that's something to keep in mind here, is that relative to baseline, we can either increase with excitation or decrease with inhibition the firing rate. And there may be something about the firing rate of the neuron that's useful for encoding information that answers these questions about how we see the world. Next up is to talk about the anatomy of the retina here. But I think this is a good point to stop it. We've already gone quite a bit here, and I want to stop it right here so that way we can absorb the key starting points, and then we'll move on to talking about the details of retinal anatomy.